Okay. And for today, we have Dr. Mercedeh Movassagh. I will go through a short introduction with you, and then she can start her presentation. Dr. Movassagh is an associate research scientist with a focus on infectious disease, computational biology, and informatics at Yale School of Medicine. She got her BS from University of Virginia, master's in biochemistry and bioinformatics from George Washington University, and PhD from University of Massachusetts Medical School in computational biology and bioinformatics. Her postdoctoral fellowship was at Harvard School of Public Health, and I had a pleasure to meet her last year at a Bioconductor conference, and I really enjoyed her talk, and I'm looking forward to hearing her today. You can take it away.
Thank you so much, Afruz, for the kind introduction, and I'm very happy to be here. I didn't really know the audience today, so I prepared some basics, but also some complicated stats and also package development information for you guys. So feel free to ask questions whenever you have questions, and we'll go from there. Let's start the screen. So is everybody seeing my screen?
Yes.
Good. Okay. So I guess here's an outline of what we're going to talk about today. Basics of what is really a package, an R package. Why do we want an R package? Then we're going to talk about what is data integration in next generation sequencing data, some of the examples using the packages that we've made, and some lessons along the way. This is going to be mostly biostats lessons or lessons for package development as we speak. So really, what is an R package? Why do we need a package? A package is a fundamental unit for shareable code. Basically, it's like a bundle of R code with some data, test sets, and documentation. And why would you need a package? So the main thing that you're going to do if you create an R package is to be able to share your code with others. And that is very important. It saves you a lot of time. And most importantly, you're going to have standardized conventions for programming and code that will lead to standardized tools. And now you're like, so why is this important? And that's very important when you're thinking about the concept of reproducibility and replicability. What do I mean by that? So I got this from Jeff Leake's talk. I think it was around 2017 he gave this talk, and this report came out in Scientific America where it talks about, is there a problem and a crisis in reproducibility of data? And the answer to that is, yeah, there is. So 70% of data generated by labs cannot be reproduced in other labs. Also, 50% of the data that is produced by one lab is not reproducible by the same lab. So that is really a big problem. And that's where software development and R packages come in. If you have tools that do statistical analysis that are the same across different scientific fields and different people can use it, then this reproducibility data is going to be minimized. And so that's why it's so important to do software development, and we're focusing in R right now. So what are the basics basically for you to be able to make an R package? And a basic R package is pretty simple to make. You just need basically 4 libraries. So you need DevTools, Roxygen, Teststat, and Knitter. Teststat and Knitter are just basically code formatting and testing your data, and DevTools and Roxygen help with package development. It's quite simple. Make sure if you're using a Mac, you have Xcode installed. If you're naming a package, it can only have letters, numbers, periods. It can start with a letter, but it shouldn't end with a period. And it's really a one-liner. So you can just write DevTools create, the path to wherever you want your package to be made, and the package name. So this is just a look of one of the packages that we've developed. It does have various components, and I'll try to talk to you a little bit about them. But the basics are the description file, the namespace, the R code, and if you're using RStudios, it will have an R project. And I'll tell you a little bit about those. So what is the R directory? Obviously, it's where your code goes. That on its own is a topic that I think a lot of people should pay attention to, because it has to do with how readable your code is and how your audience are going to be actually able to connect with your code. So you don't want to ever have one R script that has all your code in it, because that's just going to be horrible to read. People do not know what belongs to where. So ideally, you want to have R codes that are topic related separated in your package. You also don't want to have like each R script to be a function, because that's just too many and there's no connection. So you have to keep it at a sweet spot, basically. The description file really depends on where you're submitting your package. Generally, we submit our work to Bioconductor for our packages, and that has the package title, the version of the package, and that changes by every time that you upgrade your package or add something or remove something from your package. It has the information of the author, the creator, and the person who basically takes care of the package. It also has the description of the package. It seems like a simple, but you should spend some time when you're describing your package, because it has to really say what your package does. It also has to contain what sort of objects it works for and it's compatible with, and if it's doing statistics, what statistical tests it does and such. Basically, this is an introduction to your package without the user installing it and downloading it, so do spend some time on it. The license is who can basically use it and coding, and then there's the R version that it works with, and that gets updated depending on where you're submitting your what platform you're putting it on. It does have keywords. The keywords are usually depending on where it is, how your package comes up. So spend some time to think about what words you're going to use. And these are just automatically made by Roxygen. It's the libraries that your package or dependencies that your package has, the suggestions that for like, I don't know, your code or whatever that you have that you can add to that, and the vignette and where it could be found. Also, if you have bug reports, you can, there's a path to where you can basically tell us if there's a problem or report the bugs. The namespace is it's basically where you can find where your functions are coming from. So it's a way to look up the values of it's a space for names of your, how do you put that? So it's where you can find, for example, if you're using the stats, where does the function for stats come from? For example here, so I'm doing a correlation, I'm getting it from base R stats. And so the package name .rproj is if you're using RStudio, this is going to be simple because it just pulls up your project and just makes life easier. Again, so based on where you're submitting your code, there's other requirements. So there's vignettes, tests that some people use. So our packages have all that. The man that has the package information, and the news file when you're updating things is going to be on there. And so rules for package development. I talked to a couple of my colleagues who also do package development, and we really thought about what are the rules. And basically, I mean, there's a lot of little rules, but before you really want to develop a package, you want to ask yourself, are there currently packages that do the thing that I want? That's very important because you don't want to recreate the wheel. So if your answer is yes to that question, you want to ask if that package is in sync with the most common needs of users, and if it's up to date with literature and the new science found. So if you said yes, it's up to date and everything, there's really no need to develop a package. And so if your answer is yes, however, there could be improvements made to it, it could be more user-friendly, that sort of depends then. If you can really upgrade and improve the package that is already there, you will help your community. So it's suggested that you do that. And if it's not, it's very different from what is there. You think you can, with the user-friendly way of doing things, it's just very different from where there's, then yes, go ahead and develop. And so if your answer is no, really you want to ask again. So is anybody else going to be using this package in the foreseeable future or not? Is this something that I will only use, but other people will not? If the answer is no, then really you don't really need to make it unless it's for your own happiness, and go for it. And if your answer is yes, sure, go ahead and develop your package. Sorry, I thought somebody said something here. And so what does data intervention for next generation sequencing mean? So next generation sequencing data is very multidimensional. What do I mean by that? I mean that there's various platforms of next generation sequencing. There's various technologies for that. And not only that, when you get your data, it has various dimensions. It doesn't just represent your results. It presents many other things like batch effects, different sort of normalizations. If you do certain normalizations, your output is going to look different. And not only that, the biological questions or other sorts of questions that you ask about your data can be very complex. So that's what we mean when we're trying to answer a biological question. We have to integrate various forms of data to be able to do that. And an example question that we have answered is, for example, asking a simple question of, are there particular non-coding RNAs that affect the transcription of mRNAs? You have to incorporate two data sets. One is a small RNA sequencing data set, and the other is an RNA sequencing data set. Not only, I would just go back to that, not only that, but the way you ask that question is very important. So what do you mean by how do non-coding RNAs affect mRNAs? What type of non-coding RNAs? Do you mean one non-coding RNA affecting one mRNA, or do you mean a bunch of microRNAs working together to affect that? So the biological questions are very important because there's different types of statistics that you have to use to answer that question. And so another example is if somebody's doing whole genome sequencing or exome sequencing, they come up with a list of variants or nucleotide, single nucleotide polymorphisms, and they want to see if it's associated with microbial or pathogen data. Then you have to incorporate various sequencing types. Again, you have to incorporate whole genome sequencing, RNA sequencing, and 16S ribosomal RNA sequencing for the bacterial data. Even within a data set, when I say multidimensionality of one type of data set, this is an example of that. If one person asks, okay, from an RNA-seq data set, I want to identify the pathogens, and then I want to deal with the host transcripts, these are two different types of data. So picking up pathogens from RNA-seq data gives you a very zero-inflated distribution data, versus if you're looking at RNA sequencing data from the host, that's a negative binomial that you're dealing with. So incorporating those with clinical and phenotypic metadata on its own is another problem. And this is a final example that I wanted to put. It's about TCR sequencing. You're looking at T-cell receptors, and you may incorporate that data with clinical data where you're looking at, well, do cancer patients show specific TCR sequencing, like TCRs or not? And also, you might be asking about diversity, basically, of the TCRs, where you're taking into account rules of microbiome sequencing to identify diversities of TCRs. And these are the packages that we've built. I'll talk about a few of them today. Before I do that, I wanted to give an introduction for non-statisticians which I think everybody should in this crowd know a little bit. Quickly go over distributions. When we talk about Gaussian or normal distribution, it's a bell-shaped distribution. A lot of the genome-wide association studies actually have this, take this as an assumption when they're doing statistics on their data. So it's non-count data also fit to this. So one of the packages that we'll talk about, MBQTL, has the normal distribution assumptions because it deals with GWAS data. To the right, you have a log normal distribution where basically a lot of microbiome data seems to fit really well to this distribution. Paulson et al. Shows in their package MetaGenomeSeq that you can fit 16S RNA data, RNA sequencing data really well to this distribution. And I'm pretty sure you're all familiar with the negative binomial. That's a very commonly used distribution in RNA sequencing. Poisson as well. I know negative binomial DSeq uses a negative binomial assumption. Poisson, I think Macau or Macau, I'm not sure how that package is spelled out, but that has like a Poisson mixture assumption when it looks at the RNA-seq data. And for zero inflated data, those data are the ones that have really zero inflation. It's pretty self-explanatory. And so single cell RNA-seq data seems to fit well to that. Some people, like I've actually personally seen that microbiome data fits well to that distribution, and small RNA data that we're going to talk about today fits pretty well to the zero inflated data. One package that uses this for single cell sequencing is ZimWave. And so now going back to Mirtar RNA-seq, again, I'm going to remind you, this package measures the relationship of microRNAs or other non-coding RNAs with target mRNAs. I'm not going to get into the biology of this. I'll just say microRNAs are very short versus RNA is pretty long, and they target the 3' UTR of the mRNAs, the RNA transcripts, and they cause translation repression or degradation. One microRNA can potentially target thousands of mRNAs. So as you can see, that can be a problem. You don't really know what it's targeting. Now, there are algorithms that have tried to predict the possibility of this interaction, basically using the qualities, the base pair qualities of the microRNA mRNA interaction, such as folding energy, conservation, and seed region matching. And so you could use those. However, the problem, as I said, is that one of those can target thousands of mRNA. So how do you really narrow down these potentially thousands of targets? How do you know in your specific data set, what is it really targeting? So that's what Mirtar RNA-seq does. It provides statistical evidence of this microRNA mRNA interaction really happening. And so for this, you're incorporating two types of data, matching mRNA sequencing and small RNA sequencing data sets. One thing I forgot to tell you with and I'll quickly go back to this, I'm sure you're aware, but these are for models for count data. This one can work with non-count data. And the reason that is important is because when incorporating this sort of data sets, it really depends on how the data has been analyzed, like after calling for any sort of alignment, basically. It could come off as count data, or it could be an RPKM and TPM. Usually when you work with a lot of collaborators, they just want the TPM or RPKM of the data. In those cases, it's going to be hard to model those data. So you're incorporating mRNA data, which could be count, TPM, RPKM, with microRNA data, which could be count, CPM, or TPM. So technically what you're trying to do is to see if your independent variable, which is your microRNA, can predict your dependent variable or outcome variable, which is your mRNA. And you're doing this across hundreds of microRNAs and thousands of mRNAs. So what you have to do is basically pick a model that best fits this relationship. So these all could fit to a distribution. So in order to pick the appropriate distribution, you have to basically model this relationship, pick the best model using AIC, which I'll give you a little bit of information on, do some initial filtering to see if it is possible for this interaction to happen. And based on the biological question that you have, use an appropriate regression model. So it could be a univariate, multivariate interaction or sparse partial correlation. And that is if you ask, and I'll explain this a little bit more, but if you're asking what microRNA targets what mRNA, you use the direct univariate relationship regression. For multivariate, you're trying to see if two microRNAs at the same time actually have an effect on each other in terms of predicting the mRNA of target. For interaction, is the corporation of the microRNAs. And for sparse partial correlation, you assume that there is a competition basically for binding, which is sort of very accurate for binding on the 3' UTR for the microRNAs. And you could do all of those. And then
sorry, we have a question. Next.
Yeah, it's Madeline. That's my, sorry about my coughing. What sort of data comes to you as counts? What are you counting?
Transcript counts.
Per specimen, per record, or across?
Right. So the mRNA is transcript of basically genes. For microRNA, it's the transcript of microRNAs, and these are per sample.
Okay.
Cool.
And so follow up on that question, what type of samples have you used to train your model? Is this a specific type of tissue?
You could use any type of tissue. It's a very good question, actually. So for Miranda, basically, the base of can this prediction occur or not, it's in 6 different types of species. And this is all the transcripts, all the microRNA transcripts that are available for everyone to use. The 6 common species are human, mice, CMV, EBV, I think I have macaque in there as well, and one other one, KSHV, that is already pre-run. And so when you're saying what you run your model on, you run it on itself, on the data set itself, because that's the whole point of it. The whole point is basically fit a model that is best for your data set. So what we do is do this analysis of microRNA, mRNA regressions across all the data set, because they're 1-to-1 relationships or 2-by-2 relationships or 3-by-3 relationships. I'll explain it a little bit more in the next slide. But basically, you come up with AIC values, average AIC values, and the least it is better. So you pick that model to run your data set on. Is that clear?
Yes, it is. It's very good that people ask questions if it is not, because all of us are coming from different backgrounds. So it is totally okay if something is not clear for somebody. And when you ask a question, it's going to help everybody else.
Yes. Please do ask questions. So this is the first. So we recommend this on the first part to be done on 20 samples or more, because this is what gives you good statistics. For this sort of model works well if you have more samples. Now, for the situations where you have less samples, so you have time point data, what you can do is to use correlations. And that you work with fold change data, no longer count data. So for 3 or more fold changes, you basically are looking for inverse relationships between microRNAs and mRNAs across your samples. And so basically you look which one's going up, which one's going down, and this pattern continues across your samples, and you estimate a background distribution using sampling, and then you rank your p-values, and you rank this relationship, and the ones that are using F statistics, the ones that are actually significant, you basically call as significant. And of course, we do multiple testing for p-value adjustments. And then there's a third part where you're looking at 2 time points. For that, you can't really look across sample sets. What you can look at is between time point 1 and 2, what is the delta value? Basically, what is the highest inverse fold change that you're measuring? And once you do that, again, you form a background distribution estimate using sampling, and then you rank again against background distribution, and you estimate your p-value just for multiple testing. I'll show you how that will look like. It's probably going to clear things. I'm sure you're familiar with AICs and BICs. I just want to quickly, before I get to the models, tell you about them. So AIC, or archaic information criterion, which we are going to use to choose a model. Basically, the K in the parameter in the formula estimates the parameters in the model. The L is the maximized likelihood function. Lower AICs and also BICs are favorable. And then the 2K term basically penalizes when you add additional parameters. This is better to use when you're predicting something. However, if you use a BIC, it penalizes more. It's a log function, and then also it's more appropriate for finding if this is a good model or not. But because we're trying to do predictions and we're doing it across like various samples, we use AIC. And so this was what I was talking to you about when I was saying, so you use various models. In this particular case, we're doing RPKMs and TPMs. So it's not going to fit well with obviously distributions like Poisson and negative binomial, which are for count data. You can see that the Gaussian model in here is performing way better. So what I'm gonna show you is with the Gaussian model. If you're putting in count data, most likely the other ones are gonna be very helpful and they're gonna work better. And so here's an example of what you'll see. So this is a viral microRNA. It's called EBV Mir-Bart 18-3P. And this is a target mRNA, MMP7. This is a host mRNA. This is in RPKM and this is in TPM. And this is your fit, and this is one of the microRNA, this is your fit, and this is one of the microRNAs that you see that is significant and associated in terms of negative correlation if you're using a univariate model. These two are examples of your multivariate models. So you have microRNA 1 and microRNA 2. Basically, you have both of them and you're trying to see if there's a difference in the prediction when you're predicting the MMP7 gene, which is the same gene. And these two seem to be the significant ones working together. And if you're doing an interaction model, you're basically looking at the third argument of the regression, which shows cooperation. You're just multiplying it and you're seeing if these two microRNAs together are going to do a better targeting or not. And these are the functions that are used basically for this part 1 analysis. For part 2, when we're looking at correlations of microRNAs on the y-axis and the genes, and keep in mind, these are all microRNA, mRNA genes that we know the interaction can happen on. Already another filtering has been done, as I described using the Miranda predictions. And you can see that we narrowed down the targets really to 4 and 5. So this is an example of how we're narrowing down these targets. And you're looking for negative correlations. So the darker the boxes mean they have the most correlations here. And this is a function that you use for it. And for the time point example, you're looking for the biggest delta value, the biggest difference in fold change that is significant if you assume sampling and background distribution. And you see this particular microRNA right here has the highest difference in fold change if you comparison to, and while it's targeting this specific mRNAs in comparison to the other ones. So really, it depends on the question you have about the non-coding RNAs and in terms of predicting the mRNA targets. So you can look at the vignette, it's pretty well described, and feel free to always send out an email and ask questions. It has had a lot of audiences, and to my knowledge, it's the only one that is data set specific, only package that's a data specific package for microRNA analysis. Now I'm going to just completely switch gears to MBQTL, which is a package for integrating GWAS data with 16S rRNA sequencing and also RNA sequencing. And so the question that you're asking here is, what variants are associated with presence of what pathogens? Just to give you an information on what you're dealing with here is you're dealing with variants. So it's a data frame like this. You're dealing with SNPs. And then you're looking at if it is heterozygous with the value of 2. If it is 0, it's a reference allele. And if it's 1, it's a homozygous variant, basically, across your samples. This is your SNP file. And if you're looking for 16S data, you're looking at count data, basically. So this is the count metrics for the 16S data that you're dealing with. And I've already explained, these follow different distributions. The assumption for GWAS is normal distribution. So we're gonna focus on normal distribution now. So there's 3 approaches to deal with this sort of data in this package, and this is the first package that actually does this. So a lot of independent analysis has been done using the first approach and the third approach, which is a logistic regression, and the first one is a linear regression, but the second one is a novel approach that we created, and I'll explain that. But what you're looking at is basically your outcome variable as the genus, and then your variant would be basically what you have, and you're looking at across samples to see if there's an association or not. And so for the second approach, what you're doing is you're estimating the correlation between the pathogens or the taxa with each other. You're also estimating down here the relationship of your pathogens with the variants, and you're sort of adjusting for that and coming up with a row estimate. And this is very useful, and I'll show you why very soon. The third one, which is the most clean cut, is actually the logistic regression, where you assume presence or absence of a pathogen and presence and absence of a species or genus of pathogen. Okay, so let's go to approach 1. That's actually a linear regression. I'm sorry, I wrote logistic up here. So the approach 1 is actually something that is very commonly used in doing EQTL analysis. Matrix EQTL is an example of a package that uses this. This is an example of where you should not reinvent a wheel, because this package is so well written, you just need to adapt it. So we just, like, what I did was just basically write a wrapper around this and adapt it to 16S and RNA-seq pathogen calls. So again, what you're predicting is the genus and species that is associated with a specific SNP. Then you do multiple testing, correction, and you come up with an FDR. Here are the plots that are created, the p-value plots, and also expected versus observed p's that you see. For approach 2, when you're estimating row, again, so you're estimating the relationship of taxa with each other, or bacteria with each other, you can think, and you're estimating basically the best fit of your taxa and variant A versus taxa and variant 2, and you're controlling for that. That's very important because of this. So assume this heat map, these are your variants, and down here, these are your pathogens. I'll go to example of chromosome 15 because you can see another variant just 4 bases aside from it is showing exactly the same behavior as the other one. And these are variants that are in linkage disequilibrium. And you can see these are results of recombinations basically that occur. You can see that most of this down here is from a species of, a genus of Devosia. This Devosia species basically, so every genre has various species, but it's behaving like this across all of the species of this particular genre. You can see the same thing repeating various times, and it's off different species of that genre. And so it shows that this area, this particular area in the chromosome has a relationship basically with this specific bacteria. Not only that, but the bacteria seem to be having relationships with other bacteria as well. So using this method, not only you can identify communities of bacteria working together, but you can see things like linkage disequilibrium and SNPs actually behaving in the same way. So I'm guessing everybody, if anybody has questions, please speak up. And the third approach is the easy.
I think we might have a question.
Okay.
Again, how do you identify the communities?
So communities you just do, so the simplest way of identifying a community is just doing Euclidean distance. And so you can see right here, so this particular Devosia, it's of different species of this genre. You can see it multiple times. They just have a secondary name afterwards. That means they're all from the genre of this Devosia. But then Neisseria is also in there. And then Alcanovorax is also in there, and a couple of other ones. And they're all occurring at the same time. And if you look at the Euclidean distance right here, they are separated. So that seems to be a specific community that is in there. Does that make sense? Is it clear?
So that's sort of within what a topical logical group. Can you have a cross group communities also?
So cross group community, I know what you're trying to say, if you had communities that, so for example, your 16S data had already pre-assigned communities, do you see that also representing actually with the variants or not? That's a very good question. I have not tested that, but what I would assume based on the logics, for example, if you have vaginal sequencing data, there are clear communities that you see using Euclidean distance. What I assume is that you'll see a similar relationship if there was a variant associated with those communities as well. So you would see this variant is present, and all the or most of the bacteria that are within that community show a similar response, because that's the initial way of actually characterizing communities in 16S is with Euclidean distance or topic models. So you should be able to see that.
Thank you.
Sure. Okay. And quickly to the third approach. The third approach is a clean-cut logistic regression. You will have something like this. I just want you to imagine what you will have. You will have the count is basically your allele conditions if it's a 2, 0, and 1. So with the GWAS data, you'll always have the 0 and 1, 0, 1, and 2. Dependent on the heterozygosity of or not, or like being a reference on your GWAS results. And then you have a specific pathogen across a cohort of samples. You have to define 0 and 1 for that, 1 being present, 0 being absent. And dependent on the expression of that, if you're hoping to put a cutoff or not, usually with 16S, it's pretty noisy, so you should put a cutoff. depending on how the distribution looks like. What you can do is measure the relationship using logistic regression of specific presence and absence of alleles or heterozygosity of alleles with the presence and absence of a specific microbe. And then you do multiple testing and pick up the specific, the significant ones. For example, we have this significant relationship on chromosome 2 with a variant allele of A. To the right, you will see the plot that is produced if you basically specify this relationship. You will see that the GG genotype is highly associated with the presence of Neisseria meningitidis in this data set, versus the AA shows an inverse relationship. You don't see a lot of presence of the pathogen in there. So it's kind of an inverse, and you can clearly see that these genotypes are associated or not with the presence of specific microbes or pathogens. And you can use, so I just showed you 16S data, but you can have the same logic when you're doing RNA-seq pathogen detections, so you'll have viral, parasitic, or fungal data, and you can do this sort of analysis with it. So I know I said a lot. I'll quickly jump into the, well, I have to, before I jump into the next section, quickly on object-oriented programming. There's a reason why I say that, because we're talking about package development while I'm going through this as well, but just an introduction to what are objects. Basically, they're data structures that have attributes to them. The famous ones that a lot of biological data uses in R is S3 and S4 objects. S3 objects don't have a formal definition. These objects are created by setting class attributes that you can get each of these attributes by using the $sign when you're coding, and the methods belong to base generic functions, and they follow a copy on modify semantic. What that means is basically you can put an object in a function. It doesn't change that object. It just creates a copy of it. And so that really helps with performing like statistics or whatever in that function. And it doesn't change the actual object. It just creates something else, an additional fold to it. An S4 class is slightly different. That is more defined. Basically, the attributes of the objects can be accessed through the @ sign, but the other qualifications are the same. Generally, you want objects because this is a way to keep things set. You will have in gene expression data, you will have the gene expression, you will have the phenotypic data, you'll have some other normalization data, and you want to keep everything together and standardize. That's why you make them, and it's very commonly used. MR experiments are a class of S4 objects. And so I'm talking about this because a lot of microbiome and genetics data and also gene expression data are stored in this sort of objects. So when you develop a package, you have to be able to talk to other packages. So for MBQTL, I don't know if you remember, but I talked about metagenome-seq normalization method that fits really well to the microbiome data. The package should be able to work with this sort of data that metagenome-seq produces, and that's an MR experiment object, which is an S4 class. Or for Mirtar RNA-seq, it had to deal with sponge. So sponge has S3 object in there, so it had to be able to be compatible. And that brings up another issue of when you create packages, you have to make sure that they can talk to other packages so that you could continue other people's work and you can have translatability between packages. I just wanted to bring that up real quick before I move on to the next topic. This will be fast. It's just for me to show you not all packages are pure statistics, basically, or do pure analysis. Some packages, like PathSeeker, and another example I'll tell you about, are made to clean up data and do downstream analysis and make it easy for people to see results, basically, not to just do like analysis and do all these modelings and stuff. So phyloseq is an example of a very successful package for microbiome sequencing that does exactly that as well. There is no sophisticated statistics in there. It's a lot of functions that basically make the visualization and analysis of 16S data easy. And a lot of times, to be honest, when you're working with physicians, they are looking for something like that. They are looking for something that just cleans up the data and gives them the final results. So PathSeeker is a package like that as well. And you might be asking, so why do I even need to do this? A lot of us just work with RNA-seq, mRNA transcripts, or single cell data, and that you need to do a lot of processing afterwards to be able to present it. But when you're dealing with pathogen identification and microbiome data, the story is a little bit different. What you deal with is basically, this is just a heat map of a matrix of pathogens using RNA-seq. So imagine here, these are your bacteria and here are your samples. This matrix can go longer than 1,000 rows. So in reality, you should think with yourself, really, are there hundreds and thousands of bacteria, obviously dependent on tissue? This is, for example, brain, sitting in the brain. No, that's not possible, right? So basically, these are either contaminants, they're normal microbiota, or batch effects, really. So what this package does is basically try to identify what is the real signal in there, and tune out the noise, and also do some sort of operations to give you a result of what is it really there, what is differentially expressed, if any, and basically clean up your data. So PathSeeker uses two types of filtering. One of them is hard filtering using decontemp. The concept is pretty simple. It's not like deep statistics or modeling. The concept is, imagine if sequence 1 is one pathogen and sequence 2 is another pathogen. You assume that the high concentration of DNA would be associated with high frequency of that pathogen in your data set. And this is assuming every dot basically is a sample. So a real pathogen would have a situation like this. You have a lot of that particular bacteria and high amount of concentration. However, a contaminant will have a different type of behavior. And that is, if you have low levels of concentration of DNA, you'll have higher contaminants because it will have higher chances to be sequenced. It will be more in depth. You'll frequently see it. And if you have high concentrations of DNA, you'll see very little of that contaminant. And so we use this as basically our hard filtering. It will be a data set that looks like this. You'll have a contaminant column, and if it's true, you just remove that. That's the first step. The second step is a soft filtering, where you're mainly looking at the sequence features. One method of doing this is estimating a delta, looking at your control versus the actual read, and basically your real sequence, your real count of that particular pathogen would be the difference between the median of what is in the control versus what is in there actually. So that would be your real count. And then looking at things like actual read counts of the nucleotides, and if you're parsing it through a protein database, the protein database, it has a default, but the user can change it. And looking at the z-scored for alignments, because in this sort of data, both 16S and RNA-seq, you get a lot of alignments, but you have to really look at mismatches and score to the best match, basically. And so you can use that and do filtering on that. Furthermore, we just built wrappers around DSeq like phyloseq did. We additionally put Wilcoxon test that's easy to use for a user to just plug it in. If they have more than 2 phenotypes, like 3 or more, they could use other categorical tests like Korsakoff test and rank regressions to do their analysis, and we create plots for them. And finally, what really physicians are interested in is to look at the number of samples that carry specific pathogens. So plots like this at the end are going to be super helpful for them so that they know how many patients, for example, have CMV, how many of them have roseola virus or other bacterial, non-bacterial infections. We did a little bit of spatial statistics as well, and we're doing some mappings. If you have spatial information, like where the village or the city your samples are coming from across the country, we could do mapping for you in like a one-liner and tell you what pathogens are coming from which regions. And so basically this is what PathSeeker does. And I just want to, I know I told you a lot of things, so I just want to finish up with a summary. Various sequencing data require different novel or already made interventions to be combined together for you to be able to clearly explain your data and the structure of your data and basically do a good analysis on your data. And feel free to always like build packages, but generally reinventing a wheel is not a good idea. But we do need more development in biological fields because there's a lot of tailored biological questions that need specific statistics. So there's a lot of room to grow. Always model your data to make good decisions about how to do analysis on your data. And so we have another package, VDJ Dive, which I'm not going to have time to go to today. And I have other non-R packages, which I hope I'm going to actually make an R version of as soon as I find time. If you have questions or want to collaborate, here's my email, here's my Twitter handle, and here's the GitHub repo. And thank you very much. I know I told you a lot, so thank you for your patience. Any questions?
That was a great presentation. Thank you so much.
I'm glad you like it. Thank you for listening. I know there was a lot of information in there. I tried to fit a lot of things in one.
Yeah, we have some time for questions, and yeah, I think there is one question.
Yep.
So are these data separate by humans versus, Bob C mice versus ferrets, or are they combined?
So they're separate. So
Okay.
are you talking about the microRNA sequencing? Or the pathogens? So the pathogens actually that you saw, they're part of, so we're working with African data actually. And so a lot of these kids come in with infections. So in terms of infections, they are one basically. And so a part of my work is to separate the noise of pathogen from real pathogens that is making the baby sick. So that's on the pathogen and human side. But in terms of the microRNAs, some of them, yes. So if you're trying to see a relationship between a viral microRNA and a human target, yes, the same patient has the same virus in them that you're seeing the connection with. But you can just do it independently as well. So if you're a healthy person and you're interested in development, for example, you can see if microRNA A in a healthy person is affecting a target, and what target, like what mRNA. It's health. I mean, you can also look at it in a disease specific that is non-infectious. So if somebody has cancer, does one microRNA go up versus somebody who's healthy, it doesn't go up. So you can look at all these relationships depending on the really biological question that you have.
So geographical data for some of the specimens. Do you know the time sequence when this sample is taken?
Yes, I have the time sequence as well. And that's where the correlations came in. So we did an analysis for CMV infection, actually. We haven't published that data yet, but we have done analysis for the CMV infections in multiple time points, actually, to see the progression of not only the virus, but then in certain time points, because these viruses, some of them have lytic and latent phase, so they go into hiding sometimes, and sometimes they don't. If your immune system is low, they'll become reactivated, so you can see a lot of these microRNAs, or where if it's latent, you don't see them, they go into hiding. So you can actually see across time point what happens to these microRNAs if you're focused on the microRNAs. But yeah.
Thank you. Nice talk. Hard work.
Yeah, thank you so much.
Some follow-up question. Do you also have healthy individuals that matched healthy individuals, because you mentioned that they are not from here?
So for the, I'm assuming you're asking about the pathogen analysis.
Yeah.
Do we have healthy? So healthy is a definition that it's very hard to tell healthy people come and give me samples. So what we do have, though, when we're looking at the hydrocephalus cohort is some of hydrocephalus is caused by genetic defects, and some is caused by infections. So the ones that have genetic defects can really be our control in that case.
Mm-hmm. That makes sense.
Yeah.
All right, if there is no more question, thank you so much again for the great talk, and hopefully we'll have an ice cream meetup and we can see some people in person.
Yeah,
Thank you so much
for inviting me for this talk, and very excited, and all good questions.
Yeah, thank you for all the good questions. Yes. All right.
Thank you.
Bye, everybody.
Bye. Take care.